Hello, Shanta. ಹೇಗಿದ್ದೀರಾ? ಹೇಗ್ ಮಾಡ್ತಿದ್ದೀರಾ ಸ್ಟಡಿ? Good morning. Good morning, all. Let us start the class, okay? Fine. So, before going to start the class, guys, good morning, Melita. Okay, guys. So, before going to start the class, let us check whether I'm audible and screen is visible. Then we'll go to the class, okay? Fine. So, tell me, guys, whether I'm audible and screen is visible. How is your studies, Shanta? Solve more questions, okay? Whenever you are having the doubt, you can raise our teachers. Good morning, Shanta. Good morning. Okay. So let us start the class. Yeah. Okay, guys, let us start the class. You know what we are doing, right? So today we are going to continue the cell cycle, cell division, and the biomolecules. So some parts of the cell chapters are there. So that we will complete and then we will go to the next. Okay, guys? Fine. So then here is the question for you. The question is very simple and it is asked in the NEET 2013 exam. The question is, the major site of the lipid synthesis is, in the last class we have discussed, right? Okay. So tell me in the chat box, you can tell me, Shanta. So I will not launch the poll. You can answer me in the chat box. Okay, fast guys. Hi Nagveni, good morning. Fast guys, this is a revision class, no? Answer me fast in the chat box. Think that this is the exam hall. Whenever I'm asking the question, think that this is the exam hall. Okay? Answer me fast. YouTube people who are watching in the YouTube, answer me in the chat box, guys. Which is the correct answer? Think, guys. So where is the lipid synthesis is going to be taken place? Okay. So let us solve the question. In the option A, they are telling that Simplast. Guys, have you studied in the plant physiology? Simplast? No, right? It is not the correct answer. Then coming to the nucleoplasma. In the nucleoplasma, rRNA synthesis is going to be happen, not the lipid synthesis, right? So the correct for the nucleoplasma, rRNA, right, guys? So that's why option B also wrong. Then coming to the RER. So RER is the site for? Can anyone tell me? RER is a site for what? Very good, Nagvini. Yes, ma, correct. Very good. Yes. When you come to the RER, rough endoplasmic reticulum, right? So where the membrane proteins will be synthesis will be taken place here, right? So that is the reason. Option C is wrong, right, guys? Then coming to the option D, SER. Yes, in the smooth endoplasmic reticulum, lipid synthesis will take place, right, guys? Yes or no? Tell me yes or no. Yes. So lipid synthesis will take place in the RER. Not only the lipid synthesis, steroidal hormones. Steroidal hormone synthesis will take place, and also the some complex carbohydrate synthesis will take place in the smooth endoplasmic reticulum. Okay. Remember, guys. Okay. So that's why the correct reason is option D. Okay. Fine. So let us go to the very simple question. The term glycocalyx used for, okay, so it was asked in the NEET Karnataka 2013 exam. Option A, glycocalyx is present between cell wall and membrane of bacteria. Option B, it is cell wall of bacteria. Option C, Bacterial cell glyco-engineered to possess N-glycosylated protein. Option D, a layer surrounding the cell wall of bacteria. Tell me guys, which one is correct? Fast. I need answer from YouTube people. All are watching in the YouTube, answer me fast. And answer me in the Zoom also. Fast guys, fast. I'm not launching the poll. Answer me in the chat box. Which is the correct answer? Just we will eliminate which is the wrong answer. Okay. First of all, come to the option B. They are telling that it is cell wall of bacteria. Is that correct, guys? No, right. The cell wall of bacteria is peptidoglycan, right? So that's why option B is wrong, right? So it is a peptidoglycan. Peptidoglycan, right? So then coming to the option C, we have not learned about that, right? Bacterial cell glyco-engineered to possess the N-glycosylated protein. Is that possible, guys? What they are telling that in the biotechnology process, we have engineered the bacterial cell wall to produce the N-glycosylated protein. No, not right. So by naturally only it is producing the glycocalyx. We are not engineered the bacteria to produce that by the biotechnology, right, guys? So that is the reason. So that is also not the correct answer, right? Option C is not correct. We are not engineered the bacteria to produce that glycocalyx, right? Then coming to the option A, the layer present between the cell wall and membrane of bacteria. No, guys. So always the glycocalyx will be above the bacterial cell wall, right, guys? So that's why option A is wrong. Then the correct answer is no, Nagveni. So listen here. When you talk about the envelope, bacterial envelope, there are the three layers. Okay. So outer layer we will call it as a glycocalyx. Glycocalyx. Okay. Then after that another layer will come. That second layer we will call it as the cell wall. Cell wall. Cell wall is made up of the peptidoglycan. Okay. Then again the third layer will come. That the third layer will be our cell membrane, or we will call it as plasma membrane. Okay. So the glycocalyx is the outer layer of bacteria. Okay. It is made up of the carbohydrate. Okay. So where it is helps in the attachment of the bacteria to the substrate. And in the glycocalyx, if the glycocalyx in the last class also I have told you, if the glycocalyx was thick, right guys? If it is thick, we will call it as capsule, right? We will call it as a capsule. If the glycocalyx was thin, if the glycocalyx was thin means what we will call it as? Slimy layer, right? We will call it as slimy layer. Okay, guys, remember, okay? So the correct answer is option D. It is a layer surrounding the cell wall of bacteria. Okay. It is a layer surrounding the cell wall of bacteria. Okay, guys. Fine. So I think you understood this. Let us go to the next question. Which one of the following types of the plastic does not contain stored food material? Tell me. Option A, chromoplast. Option B, elioplast. Option C, alluroplast. Option D, amyloplast. Okay, guys, tell me. So it is the types of leucoplast. Okay, guys. In the leucoplast, there are the three things will come. Okay, in the options they are given. In that, which will not store the food material. The question is, understood the question? Okay. Very good, Nagveni. Very good, very good. Yes, correct. I need answer from YouTube people also. Answer me, guys, in the YouTube. Chromoplast. Right, guys? Yes, chromoplast. When you have studied over the chromoplast, guys, chromoplast in the last class, I have told you that it will store the colored pigment. Right? It will store colored pigment. For example, if you take the carotenoids, right? Carotenoids, xanthophylls, right? So chromoplast will store the carotenoids and the xanthophyll. Okay. So it will store the colored pigment. Okay. Which are those? Carotenoids. Sorry, carotenoids and xanthophyll. Xanthophyll, right? So carotenoid will give the orange color, right? If you take the carrot is orange, right? Because that is contained the carotenoids, right, guys? And you have studied about the carotenoids in the nervous system also. That is about the structure of the eye, right? That is required the carotenoid pigment, right? Retinol. Then xanthophyll, okay? And you come to the, that's why the option A is correct. Then why this option B, C, D are not correct? Let us learn about that, okay? When you comes about the elioplast will be stored lipid, right? It is a one form of the leucoplast. It will store the lipid. Okay, guys. So here it will store the lipid and it will store the starch and one will store the protein. Okay, guys, fine. So here you can see, 1 minute, yeah. Here you can see the lipid. So lipid was stored by the elioplast. Okay, 1 minute. Yeah, so lipid is stored by the elioplast. Then come to the amyloplast. Amylo means what? The thing will come to your mind. What the things will come to your mind, guys? Starch, right? Yes. So the amyloplast will store the starch. And alluro means what the things will come to your mind, guys? Seed. Seed is having the allurone layer that is rich in protein, right? Yes. So it is consisting of the protein, right? Alluroplast is consisting of the protein. It will store the protein. Okay, guys. So that's it for the question. Let us go to the concept. Let us go to the cytoskeleton. So I will ask you one question, guys. When you slept in the night, someone stole your skeletal system. What will happen? Tell me. If someone stole your skeletal system, what will happen? Tell me. Answer me in the chat box, guys. What will happen? Nagvini and Shanta, tell me, ma, what will happen if someone stole your skeletal system? Are you able to walk nicely? Normally, you will walk. Tell me, answer me in the chat box. What will happen? You can't able to move also, right? And the heart is not in that position. It will it will go to the it will move somewhere in the body, right? Yes. So the same thing happen, guys. Okay. So simply we can tell that cell is the nothing. It is having the cell organelles and all the thing. It is suspended in the nuclear. It is suspended in the cytoplasma. But there are so many organelles. So, for example, organelles are structured inside the cell with the help of cytoskeleton. Okay. Yes, guys, by the help of cytoskeleton. And one more thing you have to remember here, cytoskeletons are present only in eukaryotes. It is not present in prokaryotes. Remember, guys. Okay. Fine. So then coming to the cytoskeleton, there are three types of the cytoskeleton. Okay, which are those? Based on the thickness, we can tell. Okay, one is a microfilament, and another one is a microtubule, and third one is the intermediate filament. Okay, fine. So then coming to the microtubule nanometer was about 25 nanometer thickness. Thickness is about 25 nanometer. Okay, that's why microtubule is thicker than these two these two cytoskeleton. Then coming to the coming to the microfilament. Microfilament will be sized from 8 to 10 nanometer. 8 to 10 nanometer. Okay. Then coming to the intermediate filament will be 7 nanometer. Okay, guys. Here you can see it was not like the above the 25 also, not below the 8 also, right? So that's why it was intermediate between the microtubule and the microfilament. Okay. So that's why we will call it as intermediate filament. Okay. So in the exam, they will not ask you what is a nanometer. Maybe if they ask me, they may ask you about the microtubule nanometer. Remember that. Okay. So then let us come to the structure. What are the functions of that? Okay. So let us go through the microfilament. Okay, guys. Microfilament, each tubule of the microfilament is made up of actin protein. Remember, guys, actin protein. Actin protein. Okay, guys. And where you have studied about the actin filament? Tell me, guys. Where you have studied about the actin filament? Recall. In the sliding filament theory, actin is there, myosin is there, right, guys? Right? Yes or no? Yes or no? Tell me. You have studied or not? Yes, you have studied that, right? Yes, microfilament is helpful in the movement, right? Movement of the muscles. It is present in the muscles, guys. Okay, it is present in the muscles. 1 minute. Yeah, muscles. Very good, Nagvendi. Yes. And other than that, if you take the amoeba, okay, amoeba is the animals, right? And in the amoeba, it will come the protozoa, right? For the amoeba, it will move, right? Whenever it is moving, we will call it as cytoplasmic streaming. Okay, it will move like this, right? It will move like this. We will call it as cytoplasmic streaming. Okay, for the cytoplasmic streaming also, actin filament in the cytoskeleton, actin filament is going to be helpful. Remember, guys, where you can see the actin filament, one is in the muscle and one is in the cytoplasmic streaming, that is in the amoeba. Okay, in the lower organism, which are the organism will move by the cytoplasmic streaming. There the cytoskeleton which is helpful is the none other than our microfilament. Okay, remember. So where are the usefulness here? One is in the cytoplasmic streaming. Cytoplasmic streaming, and another one is in the muscle, in the movement of the muscle. Okay, that's it for the actin. Okay, then coming to the microtubule. Microtubule is made up of the tubulin protein. Have you seen flagella and cilia, guys? Have you seen in the lower organism? When you come to the bacteria, remember, when you're talking about the bacteria, the flagella is made up of flagellin protein. Flagellin protein. Okay. When you're talking about the eukaryotes, for example, euglena, okay, and when you're talking about the paramecia consisting of the cilia, right? So there, the flagella and cilia is made up of microtubule. Remember, guys, microtubule. Okay. So, and you have studied about the how the cell division will happen, right? How the spindle fiber will be formed, right? Spindle fiber and centriole, centrosome, right, guys? So this all the thing is made up of microtubule. Okay. Then what is the microtubule is made up of? Microtubule is made up of tubulin protein. Okay. So it is made up of tubulin protein. Which are those tubulin protein? There are two tubulin protein, guys. One we will call it as alpha tubulin and another we will call it as beta tubulin. Tubulin. Remember, tubulin protein. Okay, fine. So I have told you the microtubule nanometer was 25 nanometer and it will form the centrioles. It will form the centrioles and it will form the flagella and spindle fiber during the mitotic division and it will form the cilia. It will form the cilia. Okay, guys, remember what is about the microtubule. Okay, centriole structure is made up of microtubule. Flagella cilia, it is made up of microtubule. And spindle fiber, none other than, you know, guys, whenever the cell division was happening, if the spindle fiber was not forming, means cell division will not happen, right? So how the chromosome is going to be separate? With the help of the spindle fiber, right? So what happened here during the cell division for the cancer cell? Okay, if you take the cancer cell. Good morning, Nandish. Why are you late to the class today? For the cancer patients, guys, we will use the so many drugs, right? Vinblastine, vincristine. These are the cancer drugs which will inhibit this microtubule formation. Okay, so when it will inhibit the microtubule formation, spindle fiber does not form. If the spindle fiber does not form, can you tell me the cell division is going to be happen? Mitotic division is going to be happen. Tell me, guys. Is that mitotic division is going to be happen? Good morning, Lakan. Tell me, guys, is that I'm asking you the question. Okay, tell me, if this microtubule formation is not happening, spindle fiber not happening, is the cell division is going to be happen or not? Tell me, happen or not? You have studied already. No, right? It will not happen, right? So that is how for the cancer patient we will use this microtubule inhibiting drugs. Okay, guys, that is vinblastine and vincristine. Very good, Nandish. Never ever. Yes. So then I think you have a clarity about the microtubule now, right? Microtubule made up of two protein, tubulin, that is alpha tubulin, beta tubulin. The nanometer is 25 nanometer. Our cilia, flagella, centriole, centrosome, spindle fiber is all made up of microtubule. Okay. Very good, Sahul. Very good. Okay. Then we will talk about the intermediate filament. How many of you use the guys keratin rich shampoo? Keratin, it will smoothen the keratin, not keratin rich shampoo, sorry. Smoothen the keratin. Have you heard this thing from the shampoo advertisement, guys? It will smoothen the keratin. Have you listened to this? In the shampoo advertisement, they will give, no? How many of you heard, guys? Ma'am, we don't, or else you can tell me, ma'am, we don't have time for watching the advertisements. Okay, guys. In the advertisement, whenever the shampoo advertisement will come, they will tell that. So it will smoothen your keratin, right? So where is the keratin is present? In the hair, right? In the hair, the keratin is present. So intermediate filaments are involved in the structural purpose. Okay, guys. Structural, it is helps in the structural purposes. Okay. So here you can see provide the support for the structure and shape, provide the support for the structure and maintain the shape of the cell, and also the maintain the shape of the hair. Also in the hair keratin rich is there, and in the skin also, guys, in the skin also keratin is there. Okay, and in the nails also keratin is there. Okay, because you know that nails are very strong, right? Keratin is there because keratin is giving the providing the support, right? And in the hair, obviously hair is strong, right? And the skin is keratin is dead. Yes, pa, keratin is dead only. Okay. Have you understood this cytoskeleton, guys? Tell me, do you understood this cytoskeleton? Fast. Tell me thumbs up. Give me a thumbs up if you understood means let us go to the next. Here you can see in the diagram. See here. The cytoskeleton is there. Intermediate filament. This is the intermediate filament. Here you can see the microtubule. Here you can see the microtubule. Fine. Here you can see how all the organelles are attached and hold in the cell. Here you can see mitochondria. Mitochondria is holding, right? Here you can see mitochondria. It was holded by the cytoskeleton, right? Here this is all the cytoskeleton. There is a microtubule, right, guys? How nicely it is holding, right? Yes. Fine. Let us go to the cilia and flagella. Okay, guys. So whenever you are going to the talk about the cilia and flagella, you will talk about the 9+2 arrangement, right? Cilia, flagella is nothing but 9+2 arrangement. What is the 9+2 here? So 9 here you can see the doublet. Okay. Here you can see these are the microtubules. Those are the doublet outer side. Okay. These are the doublets. Here you can see doublet, right? How many doublets are there in the peripheral region? So 9 doublets are there. 9 doublets are there at the peripheral region. In the center, 2 doublets are there. So that's why we will call it as 9+2 arrangement. Remember, guys, in the center there are the 2. In the peripheral there are the 9. So that's why it is 9+2 arrangement. Okay. Then coming to the structure here. In the center there is a 2 arrangement. 2 microtubules are there, right? So here you can see this is a protein sheet. Okay. Protein sheet here you can see. And here, all the peripheral microtubule are attached to the central sheet by the radial spokes. Remember, guys, by the radial spokes. Okay. In the center, only one doublet now. Yes. But there are the 2 microtubules are there, right? So that's why we'll call it as 2. 2 microtubules are there in the center, right? That's why we'll call it as 2. Sorry, guys, sorry. So we will call it as 9+2. Here what we are calling as Yeah, in the center 2 is there, no? So that's why only we'll call it as 9 plus 2. In the centriole, we will see the 9 plus 0. I will tell you. Okay. So 9 is the doublet. Right, guys? And 2. In the center, you can see the doublet. 2. And it is covered by the protein sheet. And all the peripheral microtubule was attached to the central sheet by the radial spokes. And the peripheral microtubules are attached to the other microtubules by the dynein arm. By the dynein arm. Remember, guys. So this is the dynein arm. So these are the radial spokes. Okay. And you can see the it is present in the flagella. Flagella is present in the euglena, and cilia is present in the parametrium. Okay. So, and how the movement will be there? How the movement will be? Okay. When you are talking about the flagella movement, okay, guys, the flagella movement is the independent undulatory. Remember, guys, independent, right? If you take the one thread, okay, if you take one thread, if you do like this, so it will move, right? So how it is moving, like that the flagella will move. Okay. So that movement we will call it as independent undulatory movement. Independent undulatory movement. Then coming to the cilia, it will move like this. Okay. So it will move like this. So that is a stroke and anti-stroke. Stroke means it will go like this. Anti-stroke means it will come out. Okay. Stroke means like this, and anti-stroke means it will come back. Okay. So this type of the movement we will call it as rowing. Rowing. Remember, guys, cilia will show the rowing movement. Okay. Rowing movement. Rowing movement. Remember, guys, the flagella is the independent undulatory, and coming to the cilia, it is a rowing movement. Fine. So then coming to the centrosome structure. Centrosome structure, it will looks like the cartwheel appearance. Okay. So it will looks like the, it will looks like the cartwheel appearance. Okay. In the centrosome, what are the things you can see? Okay. In the centrosome, you can see, so two centrioles which are arranged perpendicularly. Okay. These are the centrioles, guys. Okay. These are the centrioles which are arranged perpendicularly. Perpendicularly. Have you seen, guys? In the centrosome will form the, here you can see the centrosome, it will form the stroke, right? So which is helpful in the forming the micro, which is form the spindle fiber, right? Yes. So here you can see how the arrangement of the centriole. Centriole I am giving here. Centriole. Okay. Centriole structure we will call it as centrosome or the centriole structure. Centrosome is different. Okay. So where two centrioles was arranged in the perpendicularly. Okay. When you are not talking about the centriole, it is 9 plus 0, 9 plus 0 triplet, 9 plus 0 triplet. Here what happened? Here the microtubules which are arranging not doublet. So there are triplet, okay? And in the center, there are no microtubules are present. In the center, we can see only the hub, which is proteinaceous in nature. Hub, remember, guys, hub, okay? So triplets are there in the peripheral region. In the center, you can see the hub. Okay, guys, fine. So this is about the centrosome. Okay. Then coming to the nucleus. You all know that nucleus is the control center of the cell. It is a double membrane organelle, right? Double membrane cellular organelle. When you're talking about the nucleus, it is made up of the nuclear envelope. In the nuclear envelope, it continues with the endoplasmic reticulum, right or not? It is continuous with the endoplasmic reticulum. On that, you can see the ribosome, right, guys? And coming to the inner membrane. So when you're talking about the inner membrane, there are the outer envelope and the inner membrane are there. And coming to the inside of the nucleus, we can see the nucleoplasma, right, guys? Nucleoplasma. Nucleoplasma is nothing but the cytoplasma which is present in the nucleus. Okay, guys. And in the nucleolus, in the nucleoplasma, we can see the DNA which is present in the chromatin-like structure. Remember, guys. So whenever it is undergoing the cell division, on that time only it will form the chromosome structure. Okay. So during the normal stage, it will be in the chromatin structure. Okay. And you can see the nucleolus, and you can see the staining like structure in the nucleus, right? Nucleolus was help in the formation of ribosomal RNA synthesis. Okay. Whereas the replication and the transcription, okay, mRNA synthesis and the DNA replication which will take place in the nucleus, okay, in the nucleus. So that is in the nucleoplasma. In the nucleoplasma, you can see the in the nucleoplasma, you can see the replication and transcription. Transcription. Right, guys? You know all, right? Protein synthesis will take place in the cytoplasma. Right. Okay. So this is about the nucleus, guys. Okay. And you know that. So in the eukaryotes, it is from the where the stones will surround around the DNA, right? So that we will call it as a chromatin structure. Chromatin, right? Chromatin is nothing but this is a DNA structure where the histone proteins are attached to this, right? DNA is negatively charged, whereas the histone proteins are positively charged. Histone proteins are very rich in arginine and the lysine, right? It will come and bind on the histone. It will come and bind on the DNA. And you know that histone octamers, right? Histone octamer will bind on the DNA and it will form the chromatin-like structure. Very good. Nuclear pore is due to the fusion of the Pa. Nuclear pore is due to the fusion of the nuclear membrane and the two membranes, outer layer and the inner layer. Yes. So how many histone proteins are there? Yes. So as you told that, so H1 we will call it as a linker, right? So and what is another one? Yes, H2A, H2B, H2A and H2B, H3 and H4. So these are one set. Okay, it will form the another set. Okay, it will form the another set, and the octamer will bind to the DNA. Whole, yes, 2 into 2. Yes, guys. Remember always H1 is a linker histone protein. Okay. Fine. This is about the nucleus, guys. So now let us go to the chromosome. In the nucleus, you have to remember genomic DNA will be present in the nucleus. Genomic DNA. Okay. Fine. So let us go to the chromosome. You all know that chromosome structure. So this is a normal chromosome, guys, before the replication. Okay. So this is a normal chromosome. Here you can see the centromere. You always remember centromere is a primary constriction, right, guys? Primary constriction. And here you can see the chromosome is having the two arms, right? So upper one we will call it as a P arm, and lower one we will call it as a Q arm. Lower one we will call it as a Q arm. Upper one is a P arm. Okay. So then fine. So in the chromosome only you can see the secondary constriction will happen like this. Okay, guys. The secondary constriction will happen like this. This thing we will call it as the secondary constriction. Okay. And secondary constriction is very rich in the satellite DNA. Satellite DNA. Remember, guys. Okay. Satellite DNA is present in the secondary constriction. Okay, guys. And here you are seeing the chromosome, right? So this is the homologous chromosome. Okay. After the S phase. After the S phase in the cell cycle, it will enter into the after the S phase, it will enter into the G2 phase, right? On that time, it is having the homologous chromosome. Okay, just the replicate, right? So it is having the just a copy of the another chromosome. So this chromosome we will call it as a sister chromatids or the homologous chromosome. Okay, guys, always remember. So where the spindle fiber is going to be attached? Can you tell me, guys, where the spindle fiber is going to be attached? Can anyone tell me? I have an option for you. Whether it is going to be attached to the centromere or whether it is going to be attached to the kinetochore. Kinetochore, very good, guys. Remember, the spindle fiber during the anaphase, the chromosome has to be separate, right? So where the spindle fiber is going to be attached? Not to the centromere, guys. Remember. So the centromere is there. Here, some protein will be there, okay? That protein we will call it as a kinetochore, okay? For the kinetochore, the spindle fiber is going to be attached. Okay, guys? And based on the position of the centromere, we have the different chromosome, okay? So that we will call it as the telocentric chromosome, okay? And where the centromere was very, it is in the, what we'll call it, in the terminal position, in the very terminal position, we will call it as a telocentric, okay? And coming to the acrocentric, so it is in the near, here you can see, so this is the centromere, here you can see it is in the terminal position. For the acrocentric chromosome, the example, can you tell me the example for the acrocentric chromosome? What is the heterologous chromosome? What is heterologous? Where it is given, Nandish? Yeah. Okay. Heterologous chromosome, it is not present part. Okay. Heterologous means if you take this is the thing means in this both the things are copy of this, right? So when it is different and when this is different, then we will call it as a heterologous. You can see that in the different chromosome. Okay. So this the sister chromatis is there, and the non-sister chromatis chromosome will come. No, that we can call it as a heterologous chromosome. Okay, but it is not present. Only homologous chromosome we will tell. Okay, fine. If the centromere was terminal position, we will call it as the acrocentric. Can you tell me Y chromosome is comes under which chromosomal structure? Can anyone tell me, guys? And submetacentric. For the submetacentric, the centromere was arranged just above the center. Okay, guys. So just above the center. Okay, if you take this is the metacentric chromosome. Okay, this is the metacentric chromosome. So here what will happen? So here the centromere was attached just above the centromere. Okay, this type of the chromosome we will call it as a submetacentric. Submetacentric. The example for the submetacentric is the X chromosome. X chromosome is the example for the submetacentric. And tell me, for the acrocentric, the chromosome example is the Y chromosome. Y chromosome is example for the acrocentric. Okay, guys. Then coming to the metacentric, you can take the chromosome number 1, 2. Those all the things are example for the metacentric, where the centromere was directly attached in the center. Okay, exactly in the center means we will call it as metacentric chromosome. Student, do you understanding or not? Give me a thumbs up if you understanding. You too, student. Give me thumbs up if you understanding or not. Fine. So let us go to the microbodies. Okay, guys. In the microbodies, we can telocentric example. Telocentric example, they are not given any example for the telocentric part. Okay. If I know, if I search, I will tell you in the next class. Okay. Fine. So then coming to the microbodies. In the microbodies, always remember, guys, if the exam in the exam is they ask which are the microbodies organelle, peroxisome and the glycoxysome. Okay. Here you talk about the peroxisome. So what is the function of the peroxisome? So it is helpful in the, here you can see hydrogen peroxide. Okay, hydrogen peroxide was broken down into the water and the oxygen in the peroxisome. Okay, guys. So remember always. Okay. So hydrogen peroxide was broken down into the water and the oxygen in the inside the peroxisome. Okay, that is the main function of the peroxisome. Okay, and also peroxisome is helps in the beta oxidation. Beta oxidation of the lipid. Lipid has to be breakdown, right? Breakdown of the lipid. Breakdown of the lipid. So that we will call it as a beta oxidation. Okay. So that will take place in the peroxisome. Okay, guys. That will take place in the peroxisome. One is the beta oxidation and conversion of the hydrogen peroxide into the water and the oxygen. Hydrogen peroxide. Hydrogen peroxide. And in the plants, guys, in the seed plants, okay, in the seed plants during the germination, this peroxisome is going to be converted into the glyoxysome. Okay, remember this peroxisome only converting into the glyoxysome, and where the lipid beta oxidation, the beta oxidation will be more in the seeds. Okay, where the seeds is containing more lipids. Okay, in that seeds, the where peroxisome is converted into the glyoxysome, and it is helps in the conversion of lipid into the carbohydrate, because for the germination we require the carbohydrate, right? So what it will do, so rapid division, rapid breakdown of the lipid will take place in the glyoxysome, and where it is converted into the carbohydrate, and it is helpful in the germination. Okay, remember. So where the lipid is converted into the carbohydrate means the beta oxidation of the fatty acid in the seeds. Okay, in the oil seeds, especially in the oil seeds. Okay, especially in the oil seeds, nothing. The peroxisome is only converted into the glyoxysome. Okay, guys, remember always. And you have to remember, peroxisome is a single membrane organelle. Peroxisome is a single membrane. It is having the single membrane lipid bilayer. Okay. And coming to the glyoxysome, glyoxysome is also consisting of the single membrane. Okay, guys. Do you understood this? Very good, Sadhu. Thank you, Lathan. Fine. So that's it, guys. If they ask about the glycosome, they will ask you that this is present in the what we will call it as the oil phase, where the peroxisome is converted into the peroxisome was converted into the glycosome. It is helped in the lipid breakdown into the carbohydrate. That's it. That's it, guys. No other question will ask from this part. Okay, fine. So let us go to the revision of the cell cycle and cell division. Okay, guys. Then here is the question for you. Spindle fiber attach on to. I need answer from you two people very fast. Okay, answer me fast, guys. Which is the correct answer? Spindle fiber is going to be attached to the. Think and tell me. Think and tell me. Very good, very good. Here only you will do the mistake, guys. Okay. So think and tell me which is the correct answer. Fast. Fast, fast, fast. Option D, right? Yes, option D. That is kinetochore, not the kinetosome. They will do the mistake like this, okay? To confuse you. Always you have to be cool when you are attending the answers in the exam. Okay, guys? The correct answer is kinetochore. Okay? Yes, kinetochore. Let us go to the next. When you're talking about the cell division, what is the cell division, guys? So it is a sequence of the events when the cell will duplicate, right? It will duplicate its organelle, like the one set of the organelles are already there, right? When it is undergoing the very good Saul, correct. When it is undergoing the cell division, it has to duplicate its cell organelle, right? So here what will happen? Cell organelle duplication will take place and the DNA duplication will take place. And finally, the cell is going to be divided into two cells, right? So that is what we will call it as the cell cycle, right? The sequence of the events one cell is undergoing. That we will call it as a cell cycle. Okay, guys. So there are two phases are there, right? So one is we will call it as interphase and another one we will call it as a M phase. Okay, guys. And you always have to remember what is the generation time. The time gap between the two cell division. The time gap between the two cell division we will call it as generation time. The generation time in the bacteria is 20 minutes. Okay, like that. So coming to the interphase, in the interphase there are three phases are there, right? G1, S phase and G2, right, guys? So here you can see the DNA content. Always remember, in the interphase, the chromosome number does not going to be changed. It will be same. Okay, you may get confusion while they ask the question in the exam. The chromosome number remains same, guys. Okay, then coming to the DNA content. DNA content is going to be double. Only DNA content. Okay, guys. Only DNA content is going to be double. Okay, guys. So here you can see 2C. And after the DNA replication, so that is in the S phase, it is going to be converted into the 4C. If they ask the question, what is the DNA content in the G2 phase? Can anyone tell me, guys? What is the DNA content in the G2 phase? Right corner part. I will explain you, Lakan. I will explain you, Pa. I will send this PPT also. Okay. Yeah. Yes. No. What I am telling you, guys. What is the DNA content? What is the DNA content in the G2 phase? 4C, right? This is the DNA content. DNA content. DNA content is the 4C, not 2. Chromosome number was 2N and DNA content was 4C. Okay, guys. So then coming to the after the, what happened in the G1 phase? In the G1 phase, the maximum all the organelles is going to be synthesized. In the coming slide, I will tell you. Okay. So then coming to the M phase. Okay, guys. In the M phase, what will happen? Karyokinesis. Karyokinesis means a nuclear division will happen. Then after that, cytokinesis, cytoplasma division will happen, right? Cell was divided into two daughter cells. Then coming to the entire process of the sequentially genetically controlled by the proteins, right, guys? So entire cell cycle process was controlled by the protein. That protein we will call it as CDK and cyclin. Remember, guys, CDK and cyclin. Okay. In the exam, they may ask you, what do you mean by CDK? CDK means cyclin dependent kinase. Cyclin dependent kinase. Remember, guys. Okay. Very good, Sahul. Very good. Correct. So then coming to the, I think if you understood this, let us go to the, it doubles 2x. Nandish, what will happen? You know, the DNA content. Very good, Shanta. Yes. DNA content, remember DNA content will be 4C, okay, and chromosome number will be same all over the phase, that is 2N, okay, fine. So let us go to the interface. In the interface, G1 phase is there, G2 phase is there, and S phase is there. So sequentially we have to come in G1 phase. G1 phase is a very longest phase, right? So what will happen, guys? In the G1 phase, the cell is metabolically active, right? Metabolically active. So what will happen in the G1 phase? All the organelles, guys, all the organelles will be double in the G1 phase, okay? And protein synthesis will happen, and the RNA synthesis will happen, and all the organelles will double except mitochondria and chloroplast. Mitochondria and chloroplast will not be double in the G1 phase. Okay, guys. So this is about, and it will take longest phase in the cell cycle. Okay, and cell size is going to be increases. Okay, so that is in the G1 phase. Then coming to the S phase, the replication will take place. Everyone know that DNA content is going to be double. Before it is 2C, now it is 4C, right? So DNA replication is going to be taken place, and histone duplication is going to be taken place, histone protein duplication, and coming to the centriole duplication. Centriole, which is helpful in the spindle fiber formation, that is also duplicate in the S phase. Okay, remember always, guys, this is a very important question. DNA replication, histone duplication, centrosome duplication will take place in the S phase. Then coming to the G2 phase, in the G2 phase is the second growth phase. In the second growth phase, what will happen? Duplication by the semi-autonomous organelles. Which are the semi-autonomous organelles, guys? None other than our mitochondria and chloroplast, right? Mitochondria and chloroplast duplication will take place in G2 phase. Remember, guys. Okay, in the exam, they may ask you, in which phase of the cell cycle that mitochondria and these chloroplast will be duplicate. You will give, ma'am told that G1 phase will be duplicate everything. No. Semi-autonomous organelles will duplicate in the G2 phase. G2 phase. In the G2 protein synthesis, na? Yes. In the G2 also protein synthesis will happen. Okay. For example, the protein synthesis which is required for the mitosis. For the mitosis, you require the spindle fiber, right? Spindle fiber protein, that is the alpha tubulin and beta tubulin, will synthesize in the G2 phase. Okay. In the G1 phase and the G2 phase, protein synthesis also takes place, guys. Okay. Fine. They are metabolically active. Okay, guys. So that is about the G2 phase. Okay. Duplication of the semi-autonomous organelle and the tubulin protein. Sorry, tubulin protein will be taken place here. Okay, guys. And so you know that mitosis consisting of the prophase, metaphase, anaphase, telophase. That is the PMAT. Right, guys? Okay. So let us go to the checkpoints. There are 3 checkpoints are there. One is between the G1 to S phase. What it will check? Whether the proteins required for the DNA replication, all the things was correctly synthesized or not. Now the DNA was correct or not. If the DNA is having any mutation, it will not send to the S phase. Okay, guys. So it will repair the DNA and then only it will send to the S phase. Okay. So here there is a one checkpoint, that is a G1 checkpoint. G1 checkpoint was not present in the G1 phase. It was present between the G1 and the S phase. Remember, okay? And coming to the G2M checkpoint, G2M checkpoint, whether the all the protein which is required for the spindle fiber was synthesized or not, and all the protein which is required for the mitosis are synthesized or not, will be checked in the G2M checkpoint. Remember, guys, G2M checkpoint. Okay. And another checkpoint is there, that is in the M checkpoint. Okay, guys. For example, ma'am, the DNA is having a mutation and some of the protein which is required for the replication was not synthesized, ma'am. Then either the cell will undergo the replication, ma'am. Never ever. It will not go to the S phase or the replication phase. It will go to the resting phase. That is the G0 phase. Okay, napa. ಅದೇನ್ ಮಾಡಿದೆ ಅಂದ್ರೆ ನೀವು ಸಿಂಥಸೈಸ್ ಏನು ರಿಪ್ಲಿಕೇಶನ್ ಬೇಕಾಗಿರೋ ಪ್ರೋಟೀನ್ ಇಲ್ಲಾನೆ ನೀನು ಹೋಗೋ ಪರಿಸ್ಥಿತಿಗೆ ಎಲ್ಲಾನು ಸಿಂಥಸೈಸ್ ಮಾಡ್ಕೊಂಬ ಆಮೇಲೆ ನಾನು ನಿನ್ನ ರಿಪ್ಲಿಕೇಶನ್ ಕಳಿಸ್ತೀನಿ ಅಂತ ಅನ್ನತ್ತಪ್ಪ. ಓಕೆನಾ? ಸೊ ಇಟ್ ವಿಲ್ ಸೆಂಡ್ ದಟ್ ನೈಸ್ಲಿ ಆಫ್ಟರ್ ದ ಆಲ್ ದ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಯು ಕಮ್ ಫಾರ್ ದ ಎಸ್ phase. Okay, so always remember, guys, in the G1 if any problem is there, means it will go to the resting phase, G0 phase. Okay, then all the thing will happen, protein synthesis will happen, then it will go to the S phase. And you have to remember some of the cell in our body, for example, heart cell. Okay, So when it is divided by the mitosis, it will not again undergo the cell division. So that's why this all will enter into the G naught phase, resting phase. Okay. Our heart cell is in the resting phase. So that is in the G naught phase. Okay. It is active, but it will not, it will growing. Okay. But it will not undergo the cell division. Remember. Okay, then coming to the mitosis. In the mitosis, there are the three phases are there. You know that, right? One is the prophase, second one is the metaphase, and third one is the anaphase, and fourth one is the telophase. What are the things will happen, guys? For example, now I have given the cell to you, right? In the cell, properly everything is there. Nucleolus is there, and all the organelles. Now only it was doubled, and everything is there, right? So how the mitosis is going to be happen now? See here, when the mitosis is going to be happen, the first in the prophase, what are the things will happen, guys? Nuclear membrane is going to be degrade. Nuclear membrane is degrade, and nucleus is there, no, in the center where the ribosomal RNA will synthesis. That is going to be disappear. Okay, that is going to be disappear, and the chromosome will move towards. Okay, it is started to from the chromatin condense. Condense means what? I have told you. Whenever the cell division is happening, on that time only it will form the chromosome structure, right? So now it is in the chromatin like structure, right? It will condense to form the chromosome. The three things will happen. Okay, chromatin material will condense, and nuclear membrane will degrade, and the nucleolus will disappear. Don't confuse between the nuclear membrane and the nucleolus. Nucleolus is in the center, right? Center of the nucleus. Okay, it is going to be disappear. Fine. So, and these things are there. What is that? Centriole. Centriole will move toward the opposite. Okay, it will move toward the opposite. Okay, guys. So now what will happen in the metaphase? If you want to study the chromosome structure and everything, for example, not structure, if you want to study if you want to study about the chromosome, means the best stage to study the chromosome was the metaphase. Remember, guys, the best phase to study the chromosome was the metaphase. What will happen here? The complete chromosome structure was happened here. Complete condensation happen here, and the chromosome will all, and the centromere always centriole move toward the opposite pole now. Okay, now the chromosome will come toward the equatorial region. They will assemble in the equatorial region. Now they are ready to divide, right, to the opposite poles, right? So they will assemble on the equatorial plane. Okay, this plate we will call it as the metaphase plate. Okay, guys, metaphase plate. Here the maximum condensation. In the exam they may ask you, the maximum condensation of the chromosome was happening in which phase? So that is none other than the metaphase. Okay, guys. So then coming to the anaphase. In the anaphase, what will happen? The spindle fibers will attach to the kinetochore, right? So already it is attached, right? So now what will happen? It will create a force. The microtubules, sorry, spindle fiber will create a force. Then chromosome will pull towards each other. Okay, pull towards the each pole. So one will go to the another pole, one will go to the another pole. Okay. So now the chromosome is equally segregated, right? So this thing we will call it as a disjunction of the chromosome. If the non-disjunction was happen, means what will happen? The ploidy, it will lead to the ploidy, right, guys? The chromosomes has to be separate correctly here. If it is results in the ploidy, which phase was result in the ploidy means that is none other than our anaphase. Anaphase, if something was happening, if the chromosome was not segregated correctly means it will lead to the non-disjunction. Okay, then it will lead to the ploidy. Okay, chromosomal disorders, you know, right? So that disorders it will read. Ma'am, structure of the interphase and the telophase are little similar now. Telophase, yes, similar. Because in the interphase, what is happening? It is undergoing the division, right? Not similar. Observe once again, not obviously pura similar. Okay. Then coming to the telophase. So what will happen in the anaphase? Only you have to remember here the chromosome will form the different structure. So that is V shape, L shape, J shape, and the I shape. Remember, guys, if you want to see the structure of the chromosome means, so that you can study the chromosome structure in the anaphase. Anaphase. Okay. Then coming to the telophase, what will happen now in the telophase? So the chromosome where the nuclear membrane, what are the things happen in the prophase? Opposite to that will happen in the telophase. So what will happen? There the nuclear membrane was degraded, right? Here the nuclear membrane will reform. There the nucleolus will disappear, right? Here the nucleolus will reappear, right? And all the organelles was disappeared in the mitosis, right? Sorry, disappeared in the prophase. Those all the organelles will reappear in those all the organelles will reappear in the telophase. Okay, guys. So now the cytokinesis will happen. Okay, cytokinesis will happen where the two daughter cells will be separate. Okay, this is what we will call it as mitotic division. Okay, guys. Lakan, which slide, pa? Can you tell me which slide, Lakan? Which slide? Checkpoint slide? Lakan, checkpoint slide or what? Checkpoint. Okay, okay, Lakan. See here. In the cell cycle, there are the three checkpoints are there. Okay. One is between the G1 phase and the S phase. Okay. So what it will check whether all the DNA was correct. Either it is not having the any mutation where the DNA replication protein like the DNA polymerase, helicase, and all the thing was synthesized or not. If it is not synthesized, then the G1 phase will not go to the S phase. The cell will not undergo the S phase. Okay. It will go to the G0 phase and it will correct and it will synthesize everything. Then it will come to the S phase. So there is a watchman is there. Okay. So that watchman name we will call it as G1S checkpoint. G1S checkpoint. Okay. So then in the S phase there is no checkpoint. Okay. Then remember in the S phase there is no checkpoint. Then coming to the G2 phase. In the G2 phase there is a one checkpoint. Okay. When it is going to the G2 to M phase. Okay. So there is a one checkpoint that is a G2M checkpoint. Okay. So what they will check in that means they will check whether the spindle fiber, right, microtubule and all the thing was whether it was synthesized or not. If it is not synthesized, means how it can undergo the mitosis, right? Most of the cancer cell, when we are going to treat the cancer cell, it is going to fail in the G2 phase only, right? So then it will not undergo the mitosis and the cell division in the cancer cell. Okay. So here you can see in the G2 phase, it will check microtubule formation happen or not. If it is not happen, it will synthesize, then it will go to the M phase. Okay. So that's why G2 M checkpoints are there only for the microtubule spindle fiber checkment. Okay. Then coming to the M phase. When it is entered into the M phase, it will check. Okay. It will check about the cytoplasma, whether all the organelles are developed or not. If the cytoplasma was in the, if it is very less means, when the cell was divided, both the cells will be very small, right? So it should be checked whether all the cytoplasma was equal to divide into the two cells. Okay. So there is a one checkpoint. That checkpoint we will call it as the M checkpoint. Okay. So there are totally three checkpoints are there. One is a G1 checkpoint, another is a S checkpoint. Sorry, another is the G2M checkpoint, and third one is a M checkpoint. In the S phase, we can't see any type of the checkpoints. Lakan, do you understood, pa? Lakan, do you understood this? Then coming to the meiosis, guys. Okay, in the meiosis, you all know that. So there are the two meiosis are there, right? Meiosis 1 and the meiosis 2. Then coming to the meiosis 1, in the meiosis 1, the prophase only containing the 5 stages, right, guys? One is a liptotin. In the liptotin, what happened? The chromosome will form the bouquet-like structure. Remember, guys, always in the leptotin stage, the chromosome will form the bouquet-like structure. Bouquet, like the bouquet. You can take the flower bouquet, right? It will form the flower bouquet-like structure. So that's why you always remember in the leptotin, the chromosome in the bouquet-like structure. Okay. So now what will happen in the zygote? Okay. In the zygote, the chromosome will come together. Okay. This is the one chromosome. This is another chromosome. It will come together for the recombination. Okay. So here in the between two them, so there is a synaptonemal complex will form. Okay. Remember, guys. So where the synaptonemal complex will form in, that is in the zygote stage. Okay. In the zygote stage, the synaptonemal complex formation will take place. Remember, in the zygote stage, the synaptonemal complex will form. Okay. Fine. So that is helpful for the synapses. Okay, guys. Synaptonemal complex. Here you can see that is the thing. Okay. Then coming to the patchitin. Okay. So coming to the patchitin, here what will happen? So here the recombination will happen. Okay. So here the chromosome recombination will happen and it will exchange the genetic material, right? Here you can see in this one chromosome, it will exchange, it will cut. Okay. It will cut first. And here you can see the ligase will join. So here you can see now this chromosome, it is having this DNA, and this chromosome, it is having this content. Okay, the recombination was happen in the Pachetin stage. Remember guys, recombination was happen in the Pachetin stage and by the recombinase enzyme. Okay, so now in the diplotin stage. Now in the diplotin stage, always remember the synaptonemal complex started to disappear. Guys. Okay, complex formation was happen in the zygote, where disappearance was started in the diplotin stage. Okay, so now what will happen? So here, when the disappearance was happen, it will form the X-like structure. Okay, so X-like structure. That X-like structure we will call it as chiasmata. Chiasmata. So many of them will get confused here. So this X-like structure we will call it as a chiasmata. Chiasmata formation will happen in the diplotin. Remember guys, in the chiasmata formation will happen in the diplotin stage. Okay, diplotin stage. Okay, and then coming to the diakinesis. In the diakinesis, what happened? The X is there, no? Chiasmata formation was happen, no? The both the chromosome will separate. Okay, guys, the both the chromosome will separate. We will call it as termination of the chiasmata. This is chiasmata, right? So here the termination will happen. So that we will call it as the chiasmata termination. I will tell you, Nandish. I will tell you. So this is what we will call it as the termination. Remember guys, in the leptotin bouquet stage, in the zygotin synaptonemal complex, in the Pachetin what will happen? Recombination will happen. Okay, recombination will happen. Then coming to the diplotin, synaptonemal complex will disappear, and in the diakinesis, termination of the chiasmata will take place. Always remember guys, this is very, very important. Then coming to the other phases. Prophase was happen, right? In the process, what are the things happen, guys? Same, similar to the mitosis only. The nuclear membrane disappear, right? Nuclear membrane was disappear, and the nuclear membrane was degrade, nucleolus was disappear, and all the organelles was disappear, right? But here in the prophase one, what happened here? The recombination was happened. That is the main thing. Always remember, in the meiosis 1 recombination happen or meiosis 2. Always in the meiosis 1. Remember guys. Okay. So then coming to the meiosis 1 is like a meiosis. Then coming to the meiosis 2 is exactly like the mitosis. Okay guys. So now what will happen? Same thing. So in the metaphase, so where the chromosome will become the very what we will call condensed and will come in the metaphase plate, and in the anaphase what will happen? It will pull, right? It will go to the each pole, and in the telophase what will happen? So it will divide it into the two things, right? It will divide into the two phase in the cytokinesis. Now you tell me whether the daughter cell here haploid or diploid. Yes, daughter cell was divided into two, right? So now you tell me these daughter cells are haploid or diploid. This is very important question, guys. So these are haploid in structure, haploid. Okay, remember. So then these two cells are there, right? Haploid cell, it will undergo the meiosis 2. Meiosis 2 is exactly like the mitosis. Okay, so the same thing happen, guys, in the meiosis 2. Nothing recombination is there, right? So what will happen? The cells will undergo the same prophase. What is that prophase in the metaphase? In the metaphase, prophase 2, what will happen? Same thing. Nuclear membrane will degrade, nucleolus will disappear, the same thing will happen. So then it will enter into both the cell will divide, and each cell will give rise to the 2-2 cell. Each cell will give rise to the 2-2 cell. That's why in the meiosis we will get the 4 haploid cell. Okay guys, yes. So what is the product of the meiosis, guys? In the meiosis we'll get the 4 haploid cell. Yes, you know that. Very good, Nandish. Thank you. Meiosis is a reductional division, right? Whereas the mitosis is the equational division. Right, guys? Mitosis is the equational division. Always remember, guys. Okay? Yes, haploid. Very good. So then coming to the... Yes, very good, Lakan. Yes, biomolecules. So here what you have to remember, guys? The metal or metabolites. So that is the intermediate. So that are in the metabolism. That is helpful in the metabolism, right? So anabolism, catabolism, you all know that. In this thing, biomolecules we will study carbohydrate, protein, lipid, and the nucleic acid, right? What do you mean by primary metabolite and what do you mean by secondary metabolite? Primary metabolites are directly involved in the growth and development of the organism, right? Whereas the secondary metabolites are not involved in the growth and development. They are just involved in the defense mechanism, right? They are just involved in the defense mechanism. So that's why we are called it as they are not directly involved in the growth and development, but they are involved in the ecological function or in the defense mechanism. Example like that, some bacteria will produce the antibiotics, right? That antibiotic will not help in the growth of bacteria, but that is helpful in the defense mechanism, right? That is the antibiotics. And the mycotoxin, the toxin from the fungi, right? You take the example where the penicillin is produced by the fungi, right? Penicillin notatum. So where it produces the toxin, so it is not led to the growth of the streptococcus, right? So it is harmful to the bacteria, right? So Nandish, what will happen here? We are concentrating on the revision on the this thing concept. In the whole one class, all the concept is difficult, right? That then also I am taking the MCQs in between. Here you can see the examples of metabolite. This is very, very important things, guys. You will get the question from this. They may ask you the question like this. Which one is the secondary metabolite act as a pigment? Or so which one the metabolite given here is example for the pigment? Remember, carotenoid. You can see the orange color, right? Carotenoid and yellow color. And you can see the purple color due to the anthocyanin. Purple color due to the anthocyanin. Okay. And alkalides, which is the secondary metabolites example for the alkalides, morphine and codeine. So these are acting as the anala painkillers. Okay, these are act as a painkillers. Okay, then coming to the terpenoids, monoterpene and the diterpene, and essential oil, lemongrass oil, and toxin, abrine and the ricin. They will ask you the question like this: which is the secondary metabolite act as a toxin? Which is that? Abrine and ricin. Then coming to the lectin. So lectin is the carbohydrate binding protein. Remember, guys, lectin is the carbohydrate binding protein. Carbohydrate binding protein. Okay, so that is the lectin. Okay, so the example is the concovalin. Concovalin is the lectin. Okay, then coming to the drugs, in the beginning only I am telling vinblastine, that is helpful in the cancer treatment. Remember, guys, this question was asked in the one NEET exam. Okay, so vinblastine is helpful in the cancer treatment, and curcumin. Curcumin is also used in the treatment of the cancer. And the polymeric substances, our rubber, gum, cellulose, these all are the polymeric substances. Okay, then coming to the enzymes. You know that enzymes are the, what are the enzymes? Biological catalysts, right, guys? So you have eaten the food that should be degraded within the afternoon, right? So who is helping in that? Enzymes, right? Enzymes are the biocatalysts, right, guys? Enzymes are the biocatalysts. Enzymes are made up of the protein, right? So enzymes are made up of the protein. What is the structure which is present in the enzymes, guys? That is the tertiary structure. Remember, tertiary structure is present. Okay, all enzymes are protein, but not all the proteins are enzymes, right, guys? If you take the hemoglobin is also protein, right? But this hemoglobin is not the enzyme, right? Remember. And coming to the enzyme, so in the exam they may ask you all the proteins are the enzyme, right? The only enzyme which is consisting the RNA, the enzyme which is consisting the RNA is ribozyme. Okay, that is helpful in the protein synthesis. That is helpful in the protein synthesis. That is a ribozyme. That is only enzyme consisting the RNA. Other than that, all the enzymes consisting of the protein. Okay, guys. Then coming to the enzymes. Enzymes are very specific about the substrate, right? Where the amylase is not going to be cleave the protein. It is going to cleave only the amylase is going to cleave only the carbohydrate, right? That is the maltose, right, guys? Remember, so those are very specific for the own substrate. They are not going to be cleave the other substrate. Okay. So I have told you ribozymes. So these are the RNA. It is behave like the enzyme. Then coming to the enzymes, they are having the tertiary structure, right? So in the structure, in the enzyme structure, where the substrate is going to be bind. Okay, where the substrate is going to be bind, that structure we will call it as a active site. Remember, guys, so this active site where the substrate is going to be bind on the enzyme. So it is like the log. Log is an enzyme, okay? So lock is the enzyme where key is the substrate. So where key is coming to bind on the whole, right? So that is the active site. Very good, very. So that's why we'll call it as a lock and key model. In the exam, they may ask you which is the lock here, which is the key here. Okay, lock is the enzyme, key is the substrate. Remember, guys, the enzyme is having the tertiary structure. So that's why what will happen whenever you are eating the food, when you boil more, okay, when you boil more, the tertiary structure it is going to be loose, right? So the bonding is going to be break, right? So due to that, what will happen? So the enzyme is going to lose its activity, right? The enzyme is going to be denature. Very good. It is going to be denature, right? So which are the bonds are present in the tertiary structure, guys? Covalent bond is present, right? So if you take the non-covalent bonds, non-covalent bond is present. Which are the non-covalent bond? Hydrogen bond is present. And if you take the disulfide bond is present. And van der Waals force of attraction is present. And electrostatic force of attraction is present. Right, guys? So these all the things are present in the tertiary structure of the enzyme. If you take the quaternary, so more than one polypeptide chain is present, right? More than one polypeptide chain is present. The example for the quaternary structure is hemoglobin, right? It is made up of the 2 beta globulin, 2 beta, whereas the 2 alpha chain and the 2 beta chain. That is the quaternary structure, right, guys? So now let us talk about the next 2 minutes. Yeah. So coming to the structure here, the enzyme. So the enzyme is going to bind to the substrate and it will form the enzyme substrate complex, right, guys? Then it will form the product. So when it is form the product, the enzyme is free now, right? So remember, guys, always if you give the only one enzyme and you are giving the 100 substrate, okay, in the always the reaction what will happen? Enzyme concentration will be less, substrate concentration will be more. If you take the take about the when you are making the curd, okay. So if you take 1 liter of the milk, you are going to inoculate only 1 spoon or 2 spoon of the curd, right? So in the curd there are the enzymes are there, right? You are taking only little amount, right? You are taking very little amount, very 1 cup, right? So which is the less now? Enzyme is very less, right? Where the substrate is more, right, guys? So when the enzymes convert the substrate into the product, again it is free now, right? It will bind to the another substrate and it will convert into the product. And it will bind to the another substrate and convert into the product. Okay, this is how the enzyme is going to be work out. Then lock and key model I have told you now only, right? Fine. So what they will tell in the lock and key model, the active site it is a lock in the enzyme and substrate fits like a key. Okay, so substrate fits like a key, but it is not true for all the enzyme sites. Okay, then we will tell that induced fit theory. Okay, so where active site is not very rigid, where the enzymes will come and bind, sorry, where the substrate will come and bind to this enzyme active site, then the structure is going to be changed. Okay, so that we will call it as induced fit theory, where the substrate will bind to the active site, then it will modify the structure. Okay, guys, that is in the induced fit theory. What they are telling in the lock and key model, the active site is so rigid. Active site is so rigid, where the substrate will come and bind to the active site. Exactly, like that they are telling. But this is not true for all the enzymes. The correct is the induced fit theory. Okay, guys, induced fit theory was given in the 1958. Now we are following the induced fit theory. Fine. Then coming to the enzyme inhibition, all of you know that enzyme inhibition. So there are the reversible inhibition are there and the irreversible inhibition are there. So in that in the reversible inhibition, there is the competitive inhibition, uncompetitive inhibition, and the non-competitive inhibition are there. Right, guys? Competitive means what will happen? So here you can see the succinate dehydrogenase is example for the competitive inhibition, right? So where the malonate will inhibit, right, guys? Malonate. So here you can see in the competitive inhibition what will happen. Inhibitor was similar to the substrate. Okay, inhibitor will similar to the substrate, where before substrate is going to be bind, the inhibitor will come and bind to this active site. So now our substrate does not get the any place to bind on the active site, right? So now what will happen? All the active site of the enzyme is going to be blocked, right? So now the enzyme cannot convert the substrate into the product. So that we will call it as competitive inhibition. The example is the succinate dehydrogenase. Succinate dehydrogenase enzyme. Dehydrogenase enzyme. Okay, fine. Whereas in the uncompetitive inhibition, what will happen? It will not bind to the active site of the enzyme, but it will bind to the allosteric site. It will bind to the allosteric site of the enzyme, where other than the active site, other than active site, it is going to be bind, and there it is inhibiting the enzyme function. Okay. For example, if you take this is the active site, here it is having the allosteric site. Okay, so it will bind to this allosteric site, and it will change the active site of the enzyme. Okay, so when it is changes the active site of the enzyme, the substrate cannot bind to this active site, right? Because the shape was changed, right? So then the substrate cannot bind, so then it is inhibiting the function of the enzyme. So that is we call it as uncompetitive. Non-competitive means what will happen? So it will not at all compete with the both the active side and in the allosteric side. So when it is binded with the when it is bind with the enzyme, sorry, when the enzyme is binded with the substrate, it will bind to the allosteric side. So thereby it is changing the function of the enzyme. Remember, non-competitive inhibitor will bind enzyme substrate complex. It will bind to the enzyme substrate complex. Thereby it is inhibiting the function of the enzyme. So here you can see in this slide. Okay, guys. So binds to the enzyme substrate complex only. Okay, fine. So then coming to the enzyme. So always remember, guys, in the enzyme. So there are the simple enzymes are there. Simple enzymes are made up of only protein. Okay, guys. Simple enzymes are made up of only protein. When you're talking about the conjugated enzyme. In the conjugated enzyme, so it is made up of the protein part. Okay, it is made up of the protein and the non-protein part. So in this conjugated enzyme, the protein part we will call it as apoenzyme. Remember, guys, the protein part we will call it as the apoenzyme. Okay, so here the non-protein part may be coenzyme. Okay, the maybe coenzyme means what? It is made up of the organic. Coenzymes are the organic molecule. Which are the organic molecule? Vitamins, right? NAD, NAD and FAD. These all act as the coenzyme, guys. Coenzymes always organic molecule. Okay. Then coming to the cofactor. Cofactors are the inorganic. Cofactors are the inorganic. Okay. In the cofactor, you can see the ions. Like you can take the iron, right? Fe. Iron is Fe, right? Then you can take the zinc, right? Then you can take the molybdenum, right? So in the nitrogenase enzyme, you can see, right? In the nitrogenase, molybdenum and iron are the cofactor, right, guys? In the nitrogenase enzyme, you can see in the nitrogen fixation, right? Nitrogenase enzyme. In the nitrogenase enzyme, you can see the cofactor molybdenum and the Fe. Remember, this is very important. Molybdenum and Fe is the cofactor in the nitrogenase enzyme. Like that. And if you take the zinc, zinc is the cofactor in the alcohol dehydrogenase. Alcohol dehydrogenase. Alcohol dehydrogenase having the cofactor, that is the Zn plus zinc. Okay, guys, fine. So this is about the thing. The protein part we will call it as the apoenzyme. Non-protein part we will call it as a cofactor or the coenzyme. If you're talking about the coenzyme, that is an organic molecule. If you are talking about the inorganic molecule, so that is a cofactor. Okay, guys. So and this is another thing, guys. So that is about the. So here only we are calling. Here only I am telling that apoenzymes are the protein part of the enzyme, and the cofactor I have told you non-protein part are associated with the enzyme called as a cofactor. And in the cofactor will be a three types. One is a prosthetic group, coenzyme, and a metal ion. Okay. When you're talking about the prosthetic group will tightly attach to the enzyme, guys. Okay. It is not like whenever the reaction was happening on the time the cofactor will bind. No, no. So here the prosthetic group was tightly attached. Okay, remember, guys. So example, EAM. Okay. So EAM part of the enzyme, that is a peroxidase enzyme. Okay. And the catalase enzyme. So that will attach very tightly. Okay. We can't separate that. Okay. Prosthetic groups are very tightly attached to the protein part. Okay. So coenzymes have told you NAD and the FAD, right? And the metal ions also I have told you, right guys? Fine. So then coming to the factors affecting the enzyme, you all know that in the beginning only I have told you temperature and the pH, right guys? So when the temperature was increasing, the enzyme activity was increasing. Okay. And all the enzymes is having the optimum temperature. Always remember, guys. Always the enzyme is having the optimum temperature and the optimum pH. Optimum pH. Okay. On that particular pH, on that particular temperature only, it is going to be work. Okay. If the temperature was very less, they are not active, right? When the temperature was very less, they are not active. And whenever the temperature was more, because they are made up of the protein, they will denature. Okay. When the temperature was less, they are not active. They are not active. Okay. When the temperature was more, they are denature. Denature. Okay, guys. So here you can see the bell-like structure. Bell-like structure. Okay. Bell-like curve. Bell-like curve. Okay, guys. Both in the 2i, it is going to be denature. And in the pH also. When the pH was less, they are inactive. When the pH was very high, it is going to be inactivate the enzyme. It is interfere with the enzyme structure, and it is going to be modulate the active site of the enzyme, and it is going to be make the enzyme not active. Okay, guys. Then coming to the enzyme concentration or the substrate concentration. Here you can see when the enzyme concentration increasing, the reaction is going to be increases. Okay. And here you can see increases in the enzyme concentration, increases in the rate of reaction. Okay, guys. But this graph is very, very important. That is a substrate concentration. So this curve, we will call it as hyperbolic curve. Hyperbolic curve. Okay, guys, we will call it as a hyperbolic curve. What will happen here? When you increase the substrate concentration, so the reaction is going to be increases. When you goes on increasing the substrate concentration, goes on increasing the rate of the reaction. At particular time, what will happen? The enzyme become the saturated. Okay, the enzymes become the saturated. It cannot able to convert the substrate into the product. Okay, so on that time, it will reaches the maximum stage that we will call it as the, here you can see, so maximum velocity. Okay, sorry, what it is reaching here you can see. So we will call it as a hyperbolic curve. Okay, then it is not going to be increases. Okay, it is going to give you the maximum velocity, V max. We will call it as this V max, maximum velocity, V max. Okay, guys. So here on the x-axis substrate concentration are there. On the y-axis rate of the reaction is there. Okay. So the curve is hyperbolic. Okay, guys. I think you understood, guys. So if you understood this concept, please give me a thumbs up. That's it, guys. That's it, guys. Fine, Lakan. Fine. Thank you. I'm ending the class. That's it. Thank you so much. Whenever you free, watch this in the YouTube also. This is there. Okay, when you're free, watch this again. You will come to know that. Okay, guys, read this chapter and solve more questions. All the best for your exam preparation. Bye, guys. Take care. Nandish, we are taking only the important concept of the biomolecules. Okay, so those are very important. Where the so many questions was asked in that concept. In all the crash course, we are taking very, very important topic where they are having the very high weightage. Okay, very high weightage. So those are the things you can study. Yes. Okay. Yeah, you can leave. Fine, guys. Thank you so much. We will tell about the inform or the all ticket. Next chapter, genetics. Genetics. Molecular basis of the inheritance. We are going to study genetics and the molecular basis of inheritance. Fine, Nandish. Yeah, I will do physiology also, Nandish. But start reading, okay? Start reading because this is a crash course. I have to go fast. So start reading those all the chapter. When you pre-prepared and come, then it will be a very good for you. Thank you, guys. If you have any question, you can ask me. You can leave the class, guys.